In this video, we're going to cover these learning objectives, and really everything here surrounds this term osmosis. So if we get comfortable with osmosis, then these other things become pretty simple and straightforward. So what is osmosis? Let's define that. Osmosis is the movement of water. And so it's strictly used when water moves. That's it. So what I wrote here is a very kind of loosey-goosey definition of osmosis, which is diffusion of water. And in parentheses, I said kind of, because it's not technically diffusion of water. Water doesn't move by diffusion. It moves by osmosis. But the concept here is important. And so let me explain. And then we'll do this other definition. If we have a cup here, and I have this barrier, semi-permeable membrane, and I have chamber A on the left side, chamber B on the right side. So if you look at that, you could say, well, which one has a higher concentration of solutes? And then in this case, the solute is the thing that's dissolved in water. And in this case, let's just call it salt. Okay, so these little dots are salt, and the rest of that purpleness, let's just call it water. I know it's purple or pink, but whatever, let's just call it water. So where do I have a higher concentration of salt? Well, that will be in chamber B. Now, if we were showing you this picture in our last lecture, I would say to you, okay, well, which way does salt want to move? Does salt want to move from chamber A into chamber B? Or does salt want to move from chamber B then into chamber A? And you would say, well, salt is going to move according to his concentration. He wants to go from chamber B into chamber A. This is the way salt wants to move. But today's lecture, we're actually talking about osmosis. It's something different. So I'm going to put some conditions upon this. I'm going to say that this membrane is actually not permeable to salt. It's just for whatever reason, the way the membrane is set up, salt cannot move. Okay, salt wants to move from B to A, but salt cannot move from B to A. Let's just pretend that this membrane has a bunch of holes in it, but the holes are way too small for salt to make its way through. Okay, but water is actually much smaller. So what I want you to see then is that water also wants to move. So that tendency for water to diffuse from areas of high concentration of water, which is chamber A, to low concentrations of water, which is chamber B, water will therefore want to move from chamber A into chamber B. Now, you can't see me making these little air quotes, but what I'm doing is when I say high concentration of water, we're actually not allowed to say that because the convention is when we talk concentration, it's about solute. And then the solvent, which is the thing that the solute is dissolved in, is always going to be water in the case of human biology. And so here, because the term concentration is really reserved for solutes and not for water, we describe things a little bit differently. That's why I said we can't describe osmosis as diffusion of water. Even though conceptually it's the same idea, technically we can't say that. So what is the real definition of osmosis then? The real definition of osmosis, the one that you're allowed to say is this. It's the movement of water from areas of low solute concentration to areas of high solute concentration. So in this, A has low solute concentration, B has high solute concentration. So which way does water want to move? Water wants to move from chamber A into chamber B. And so that's in fact what we see. Water will go from chamber A into chamber B, and now that's what we see. There's a greater volume of water in chamber B. Now ultimately, if we take a step back, we can think of it this way. When salt wants to diffuse from chamber B to chamber A, remember that it can't, but it wants to. What's really happening? Salt is kind of moving in a way so that the saltiness is the same in chamber A and chamber B, right? Isn't that what's really happening? Some of the salt in chamber B is going to diffuse over to chamber A so that the saltiness level is about the same. So that's how diffusion ends up working. It ends up working, and things will move until there's no more gradient, until there's no more difference in concentration. Well, water's no different. When water moves here, what is it doing? It's moving from areas of low solute concentration to high solute concentration. And ultimately what's happening? The saltiness level, so to speak, or the amount of water to salt ratio is the same in chamber A and chamber B. That's what's happening. So, again, what is osmosis? It's the movement of water from areas of low solute concentration to high solute concentration. To be honest, this definition, you need to memorize it. Just memorize it and then make it make sense as you go along. You will see diffusion all over the place. We will see osmosis all over the place through the rest of Bio 141 as well as Bio 142. So memorize the definition for now if you have to, but try to make this make sense where there's kind of an equalization of water to salt or water to solute ratios on both sides. So the next question I'm going to ask you is osmosis. Do you need energy? Is this an active process or is it passive, meaning there's no energy required? Passive would mean that there's no energy required. And the answer is it's passive. Water's just going to move that way. This is talking about the same thing. I've got here like a weird fancy tube like this, and again, a selectively permeable membrane. And I'm going to call this chamber A, I'm going to call this chamber B. And what we have here is that these solutes, these gold little things, are in chamber B. Which way do these things want to move? Well, you know which way they want to move. They want to move from B to A, but they can't do it because this membrane is selectively permeable and it happens not to select for these little gold triangles. So what do we see then instead? Water ends up moving because water will move from areas of low solute concentration to high solute concentration. A has low solute concentration, solute meaning this little gold guy. B has high solute concentration, and so water moves from chamber A to chamber B to the point where it even fills up chamber B to a greater extent. Now, this idea of osmotic pressure, you need to be aware that we're going to use that term osmotic pressure. Actually, to be honest, we're not going to use it until Bio 142, so maybe I should just leave it out for now. But understand that when water moves, if I had a little plunger here, if I had a little thing to stop that, when that water moves, it's going to exert a certain amount of pressure. Because of that drive to move in that direction because of osmosis, it exerts a certain amount of pressure, and that is the osmotic pressure. So by definition, the pressure that I need to apply here in order to equalize these levels, that applied pressure is the osmotic pressure. But my point is you can actually quantify osmotic pressure. And again, we'll use that more at length when we talk about tissue level exchange, where blood delivers solutes and water into your capillaries to get to your muscles and brain and stuff. But again, that's all for another semester. This is saying the exact same thing. I'm not going to go through it because it's really saying the exact same thing as the last two slides. So pause the video and take a look at the slide or pull it up on your PowerPoint. But you'll see that really they should say the same thing. The only term that I want to throw out there right now is this term osmolarity. So that term osmolarity is right over there where my blue arrow is pointing. And there it is again, there it is again, so there it is again. So osmolarity is just a fancy word to say concentration of something. So that's all osmolarity is. So you'll hear me say osmolarity of a solution. So in other words, if I were to say, hey, chamber A or chamber B, which one has a higher osmolarity? It means, which one has a higher concentration. And concentration of what? Well, in this case, it's got to be these little circles. That's the only thing there. And so I'd say, oh, chamber B has a higher osmolarity than chamber A. Same thing over here. Chamber B has a higher osmolarity than chamber A. That's so just get comfortable with the term osmolarity. It's just another way to say concentration. Okay, so now let's see. We've covered osmosis, and now let's talk about these terms: hypertonic, isotonic, and hypotonic. Well, to be clear, what we need to start off with are these prefixes. You probably know what hyper means. It means greater than. Iso actually means the same. And hypo means less. So, for example, hypertension you've heard of, which is high blood pressure. Well, there's something called hypotension, which is low blood pressure. So hyper is a prefix meaning higher than or greater than. Iso means same, and hypo means less than. So what the terms mean. Then you have to know what tonicity means. So tonicity is another term that we use when we compare concentrations of something to yourself. So let me give you an example. This right here is let's just call it your red blood cell. This is the inside of your red blood cell, and this is the outside of your red blood cell. And you see that there's a whole bunch of these little black dots, which we're going to call solutes. Whether we call them glucose or sodium or whatever, it's really irrelevant. They're just some generic solute for now. If that sounds too weird for you, then let's just assume it's salt. Okay, it's just salt. So you have a certain amount of salt on the inside and a certain amount of salt on the outside. Now, if you were to count dots, 1, 2, 3, 4, 5, 6, 7, 8, I have 8 dots on the inside, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, whereas I have 11 dots on the outside. So I have more dots on the outside, but I've got greater volume of stuff on the outside. To say it differently, if you were to take a spoon and scoop out a little bit of this water on the inside and taste it, it would have a certain saltiness. If you were to take a spoon and dip into this water and take a sip of it, it would have the same level of saltiness. So what I want you to see when you eyeball this thing is that the concentration or the saltiness or the osmolarity is roughly the same on the inside and the outside. We describe this outside as a hypertonic solution. It is a hypertonic solution. Hyper means greater than. Greater than what? Well, the reference is U. Okay, the reference is U. So this is your tonicity, so to speak. And a hyper, did I say it was going to be hypertonic? I said that completely wrong. This, because the saltiness is the same, is an isotonic solution. So you may have to rewind that video. I may have misspoken. This particular slide, because the saltiness is the same, we're going to call it an isotonic solution. It's the same saltiness. Now, let's just go in contrast with this. You have the same amount of salt on the inside as you did in the previous slide, but a lot more on the outside. So which water is saltier, the outside water or the inside water? And you should say the outside water looks like it's going to be saltier. So therefore, this is a hypertonic solution. So one more time, in case I did misspeak, this, because the saltiness is the same, is a isotonic solution. This, because the saltiness is greater on the outside than my reference point, the reference being you and your cell. This is a hypertonic solution. And then this right here, because the outside is going to be less salty than the reference point, which is you, this is called a hypotonic solution. And just to make absolutely clear, because again, I hate to misspeak if you guys are going to just forget that, this is going to be called an isotonic solution. And then, here, let me just copy and paste that. This over here, what we're going to see here is actually going to be called a hypotonic solution. And then this over here, we're going to refer to this as a hypertonic solution. Okay, so isotonic, hypotonic, hypertonic. Go back and watch that last 60 seconds again if you need to. Okay, so the question then becomes, now we know what hypertonic, isotonic, hypotonic mean. What happens if a red blood cell is placed in each of these solutions? Well, here's what's going to end up happening. I'm going to use the same pictures to show you. In this situation, we're just going to tell you for now that it's going to be water that ends up moving. For now, let's just assume that the solutes on the inside cannot go on the inside, or cannot go out, and they cannot come in. Remember this whole selective permeability thing that I showed you in the previous slide? Assume that our cell membrane right now is selectively permeable, and none of these things are going to move. None of the things that I'm showing you right here. So the only thing that can move is water. In this situation, which way does water move? And you might say, well, there may be a little bit of water coming in, and you know what? There may be a little bit of water, similarly, that is going out. But in the big scheme of things, there is no net movement of water, and so that's it. So what's your answer to the question? What will happen to a red blood cell when it is placed in an isotonic solution? And your answer is nothing. Nothing happens at all. There is no net movement of water. Okay? So now we're going to do the same thing with the other solutions. What happens if I place it in a hypotonic solution? Well, again, let me just borrow one of these arrows. Which way does water want to move? Water wants to move from areas of low solute concentration to areas of high solute concentration. So what we're going to see here, we're going to see water will move from the outside to the inside of the cell. Now, what I want you to think of, though, is that this cell has just a limited capacity for water, and as you fill it up more and more, that cell is going to swell up, and the cell membrane is going to start bulging with all this water on the inside. Now, you know why water wants to move. It wants to move because there's low concentration on the outside of solute, high concentration on the inside. But what ends up happening is water ends up having a net movement from outside to inside the cell. So to go back here, in a hypotonic solution, what happens? Water enters the cell in the big picture. Lastly, in a hypertonic solution, which way does water move? Well, water goes from areas of low to high solute concentration. In this case, water will exit the cell. You see that right over here. Water will exit the cell, and that essentially dehydrates the cell. So what we're going to say in a hypertonic solution, water exits the cell. Last slide to show you is this. Here's my isotonic solution. A little bit of water comes in, a little bit of water comes out. Everything's even. Hypotonic solution, there's a lot of water that comes in, and there may be a little bit of water coming out as well too. Water is always sloshing around, so that's why we talk about net movement, means overall movement. So there's a net movement of water into the cell. The cell swells up like this. Here it's got this nice clean shape. Here it starts swelling up like a big fat balloon, and then eventually it could pop. And if it pops, we call that hemolysis. Lysis, you've heard that term before, means to break. Remember, glycolysis was breaking glucose. Lipolysis was breaking fat. Hemolysis is breaking red blood cells. Now, in a hypertonic solution, what do we see? Well, I showed you here, there's going to be exiting of water. And again, you're going to see, well, in the big picture, a bunch of water leaves. There may be some coming in, but the net movement is for water to go out. So when that happens, the cell itself shrivels up because water is leaving it. And that process, we give a fancy name called crenation. So to answer the question, what happens to red blood cells? A hypertonic solution, water exits the cells, and potential hemolysis. Okay? Isotonic solution, nothing happens. Hypotonic solution, the overall effect on the cell is for crenation to occur. And that's it. So hopefully this made sense to you. If it didn't, I'm going to encourage you to go back and actually, to be honest, watch it again. Because these ideas of concentration for the previous lecture and osmosis in this current lecture, these ideas are really so important that if you don't get this now, then you'll probably be okay for the rest of this unit. But as we get into muscles, you're really going to get messed up a lot when we start talking about electrical properties of a muscle. When we get into nervous system and looking at electrical properties and action, potential, conduction, you're going to get really messed up. When we get into Bio 142 and look at the heart, you're going to get messed up. When we look at the gut and the kidneys, you're going to get messed up. So it's really important that you have a mastery over these terms right now. So mastery over terms like osmosis, mastery over terms like diffusion, facilitated fusion, primary active transport, secondary active transport, which I realized was from a previous video, but terms like concentration gradient, things going down their gradient, things going against their gradient. All these things are really, really critical for you to understand physiology. Okay, that'll wrap it up.